In Chapter 12, we get to take a look at some of the science we talked about in Chapter 11 and how we as humans use that to modify organisms. We begin with this idea of biotechnology, and within biotechnology, we'll start with genetic engineering. Genetic engineering is the modification of the genes that are found in an organism. This can be done in different ways. The first way that we did this as humans was the creation of transgenic organisms. Transgenic organisms are organisms that have a gene that came from another organism. A good example would be the picture that we have on the lower left-hand side of this slide. These fish are all glowing. They're glowing in different colors. But the first ones that we created, the first glowing fish we created, are shown in the very lower left-hand side of this picture. These fish are referred to as glowfish. They're actually patented under that name. And they are fish that possess a gene called the pGlow protein. This gene occurs in jellyfish. It makes a certain species of jellyfish glow under normal conditions. We never modified the jellyfish, but we removed the gene from the jellyfish, placed it into the fish, and the fish glow. The fish that are glowing other colors are fish that have other genes in them that make them glow in different colors. And so, for example, we look over here to the right. This is a puppy, a beagle puppy, that glows red. This particular puppy emits red fluorescence as a result of the fact that it has a gene in it that comes from coral that makes this coral glow. So transgenic organisms are one good example of genetic engineering. This is possible. Genetic engineering is possible. The creation of transgenic organisms specifically is possible because the genetic code is universal. As we've talked about before, there's no difference in the DNA in the jellyfish, the fish, the dog, humans, plants. The genetic code is universal, and so we can easily transfer genes from one organism to another. Cloning genes refers to the process of making identical copies of these genes, and we'll talk about cloning genes next. So, interestingly, what we see is that recombinant DNA is created via this process of cutting DNA and gluing it back together. Now, oftentimes we use a vector to transfer DNA from one place to another. A vector could be a virus, or a vector in this particular case is a plasmid that comes from a bacteria. If you think back to our discussions in lecture, you'll recall that we talked about plasmids previously. Plasmids are small rings of DNA that contain 10 to 100 genes, and they are essentially extra DNA that's found inside the bacteria. You have the bacterial chromosome, which is a very large loop, as you'll recall, but additionally, inside the bacterium, you also have this little ring of DNA that's known as a plasmid. Now, when we make recombinant DNA, then we use these enzymes referred to as restriction enzymes. Restriction enzymes could be thought of as molecular scissors that cut DNA at particular locations. For instance, in this diagram from our text, we have a sequence of DNA and we see AATT. And we have a particular restriction enzyme that cuts the DNA whenever it sees the sequence AATT. The restriction enzyme comes along and cuts the DNA. But when it cuts the DNA, it doesn't cut it, if you follow the cursor here, with a straight slice through the DNA, but it cuts it in this uneven fashion. And so the cut in the upper sugar phosphate backbone is made here, but the cut in the lower sugar phosphate backbone is made here. This leaves us with this overhang, this AATT there and TTAA there. We refer to this overhang as sticky ends. What this means is that if we use the same restriction enzyme to cut, for instance, DNA from a cow, if the cow DNA is on the right, and DNA from a bacteria, if the bacterium DNA is on the left, then we can recombine the DNA from the cow here and the bacterium here because of this overhang, these so-called sticky ends. The DNA will spontaneously glue itself back together. And if we provide the enzyme DNA ligase that you remember from chapter 11, that can seal the sugar phosphate backbone, then we have recombinant DNA. We have DNA from two species that have been recombined as a result of two things: the sticky ends that are created by restriction enzymes, and the enzyme DNA ligase, which seals the sugar phosphate backbone. Now, here we have an example of this process being conducted. So we have a human cell here, and we have the gene for human insulin found on the human chromosome. So we use restriction enzymes to cut out this gene for human insulin. And here we have the gene for insulin. On the right-hand side of this diagram from our text, we have a bacterium, which is this entire oval shown here. Inside it, we have the plasmid, aplasmid that's found inside this bacterium. We use the same restriction enzymes to cut the plasmid. And then, because of the nature of sticky ends that we talked about on the previous slide, we can glue the human DNA into the bacterial plasmid, the bacterial DNA, and we end up with a recombinant plasmid. So the green DNA represents bacterial DNA. The red DNA represents the red DNA represents human DNA, but it's all combined together. I mean, there's really no difference in the DNA. It's just that we know the red DNA, in other words, the gene for insulin, came from humans, and the rest of the plasmid came from a bacterium. The advantage to doing this is illustrated in the remainder of this picture that comes from our text. So what we can do is take this plasmid, place it back into a bacterial cell, and that bacterial cell then divides. Once the bacterial cell divides, then it's going to make copies of its own chromosome, which is not shown here, but it's also going to copy the plasmid. And so you can set this up one evening and come back in the next morning, and you will have millions of copies of that bacterial cell. Each bacterial cell will have a copy of the plasmid. Now, interestingly, the bacteria doesn't know that this red DNA is human DNA. It just sees it as DNA, and so it's going to produce whatever product is specified by that DNA. In this particular case, the bacterium is producing human insulin, and so these little blue dots or circles are supposed to represent insulin, which is interestingly a protein hormone that is produced, of course, in all humans. But if you're a diabetic, then you have to take insulin. At least it's a type 1 diabetic. You have to take insulin in order for your body to function properly. And if you take insulin or know someone that takes insulin, this is the process by which insulin is created. All insulin that's taken, and sustains a huge percentage of our population that are diabetics, is created via this transgenic process. Now we can ask the question, is there any difference in insulin created by a bacterium versus insulin produced by a human, so-called natural insulin? And the answer is no. Those two insulins would be absolutely indistinguishable as a result of the fact that it's a human gene, it's human DNA that's being produced by the bacterium, and that insulin is exactly the same as insulin that would be produced in the human's body. Now, your book is illustrating that there's two purposes, two reasons to do the procedure that we have just described. One is what we just talked about, making human insulin. The other is that we have cloned a gene. We mentioned a few moments ago that the process of cloning a gene. This is completely different than the use of the word cloning in terms of cloning an organism. So in this case, we've cloned a gene. We can cut out this gene, and then we can use that gene for any purpose that we desire. So one common thing is to send this gene off to be sequenced. That's something we commonly do in our research. It's also possible that you would want to take this gene and insert it into another cell type for one reason or another. But whatever the case, you have cloned this gene. So you've taken the gene, you placed it into a bacteria. The bacteria has made 1 million copies of this gene. You can now cut the gene out and do whatever it is that you need to do with that particular gene. We now begin another topic in our biotechnology chapter, the topic of PCR, the polymerase chain reaction. The polymerase chain reaction is one of the biggest discoveries in science in the past quite a number of decades, it's really changed the world significantly. And what PCR does is the following. It allows us to copy DNA. It allows us to make many, many copies of a particular sequence of DNA. You all have grown up in a world in which there was always DNA evidence. You watch TV and you watch a movie and you see DNA evidence introduced into a trial setting, and you don't think anything about it. But interestingly, the ability to use DNA as evidence is the result of PCR. Before PCR, there was no DNA evidence in forensic settings, at least almost always. In rare cases, you could have enough tissue just to look at DNA without amplifying it, but nearly always, when you are using DNA in a forensic setting, you're having to copy it using PCR. The author that we see on the screen in the upper right-hand side there is Kary Mullis. Kary Mullis is the scientist that invented PCR and received the Nobel Prize for that invention. PCR came along in the early 1990s. The first O.J. Simpson trial occurred in the summer of 1995, and PCR and therefore DNA evidence in a forensic setting was so new in that trial that the O.J. Simpson defense team called Kary Mullis, the Nobel Prize-winning scientist who invented this, as a witness. That would never be done today because we're so accustomed to and so accepting of PCR. But it was so new at that time that, interestingly, he was called as a witness in that setting. So keep in mind, this doesn't go back that far. I realize you guys are born after that time, but not that far after that time. So mid-1990s was the origin of PCR. It's worth noting that all the viral testing that's being done now, all of that is accomplished via PCR. And so without PCR, we wouldn't have, the testing is essential in limiting the spread of the virus, as you're hearing over and over again now. But without PCR, there is no testing. One of my recent research students was hired. She hasn't graduated yet, but she was hired by a lab in Lexington. They were scrambling to find anyone that they could that knew how to do PCR, because, as you would imagine, of the huge need for this huge influx of testing that they suddenly had. So DNA polymerase makes DNA. It copies DNA many, many times. And interestingly, what we see is the following, that the DNA polymerase that's used in PCR, so we want to make sure we have our terms correct here. PCR, the polymerase chain reaction, is this reaction that copies DNA. So the copying that's done in PCR is done by DNA polymerase. The DNA polymerase used in PCR comes from a bacterium known as Thermus aquaticus. The genus and species name is Thermus aquaticus. Why is that the case? Because Thermus aquaticus is a bacterium that resides in hot springs in Yellowstone National Park and possibly other places in the western United States. But it's resistant. It lives at very, very high temperatures in those hot pools, and therefore the enzymes that it possesses are resistant to degradation or to damage at high temperatures. This means that in PCR you can heat the DNA, and when you heat the DNA, you don't destroy the DNA polymerase. If we used human DNA polymerase, when we heated the DNA, we would destroy the DNA polymerase. Interestingly, what we see is this, that we use primers in PCR because primers pick out which segment of DNA is going to be amplified. As you know, your genome consists of 3.2 billion bases. When you use PCR, you're only amplifying a very small fraction of the genome, and it is primers that determine which part of the genome are going to be amplified. You're not amplifying the entire genome, just a little segment, and primers will bookend. In other words, they'll determine where's the start of the sequence you're going to copy and where's the end of the sequence you're going to copy. If we look at this picture, we see a nice illustration of what happens in PCR. Here is our original strand of DNA that we're going to copy. We place this DNA into into a little PCR tube, and then we place that tube into a PCR machine. The PCR machine is going to heat up the DNA to 95 degrees, and you see when you heat up the DNA, then you denature, we call it denature the DNA. The two strands of the double helix split. We then cool the DNA down. We allow the primers, shown in green there, to bind. The primers are going to tell us what section of DNA is going to be amplified. And then we heat the DNA up a little bit to 72 degrees. The annealing temperature is likely to be 60 degrees or so. We heat the DNA up to 72 degrees, and we copy the two strands of DNA using our DNA polymerase. So the bottom line is, after one cycle, we have doubled the amount of DNA that we have. After 2 cycles, we've quadrupled it. After 3 cycles, we have 8 copies of the DNA. So in other words, with each cycle, we double the amount of DNA present. And the bottom line is that when you run 30 cycles or so, you have millions of copies of that DNA segment, and you're able to actually see that particular DNA segment. Now, this leads us to the next step in visualizing DNA. And so this is something that we did in lab before spring break. And what we see is that we use gel electrophoresis to visualize DNA. So we have to copy the DNA using the technique that we just described, and then we put it on a gel, we turn the electricity on, and the DNA migrates through the gel. DNA has a slight negative charge, and so it moves from the negative side of the gel, which I'm illustrating here with my pointer, towards the positive side of the gel. DNA migrates through the gel because of the charge. And what we see is the following. We have DNA from the crime scene, DNA from suspect A, and DNA from suspect B. When we conduct DNA fingerprinting, we use these little regions of DNA that are essentially mutations. They are places in the DNA where the DNA polymerase has made a mistake and has, a lot of times we refer to it as stuttering. So what it means is this, that for instance, suspect A here, on his 2 chromosomes, each chromosome has 12 of these stuttered repeats. So the repeats would be very short. They might be ATC, for instance, just 3 bases. So in 12 repeats, you have ATC 12 times, ATC, 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 ATC. And in the case of suspect B, on one chromosome, he has 12 repeats, and on the other chromosome, he has 16 repeats. He has a larger segment there. These segments are copied using PCR. We place the DNA into a gel, and we turn the electricity on. The DNA migrates through the gel from the negative side of the gel to the positive side of the gel, but the speed at which the DNA migrates is determined by the overall size of the DNA. Smaller pieces of DNA migrate faster because they can make their way through the gel at a more rapid pace. This means when we look at suspect A, both of his repeats are fairly small, and so remember we're starting here and moving down in this direction. So he has one band, B-A-N-D, of DNA that moved fairly far through the gel. If we look at suspect B, he has 2 bands. He has the 12 repeat band that moved a fair distance, so that of course represents that chromosome, but he also has a 16 16 repeat band shown here. That band is bigger, and so it didn't move quite as far through the gel as a result of its size. 16 repeats here, 12 repeats there. We also have DNA extracted from the crime scene. Of course, we see that we have 2 bands there, a 16 repeat band and a 12 repeat band, and so that matches up nicely with suspect B. And this is a DNA fingerprint. DNA fingerprints are very simply a process by which the DNA is copied and then run through a gel, and then we get an image of that. Now, this is, of course, a simplified one, similar to the one that we did in lab, because there's only 2 locations that are used here. You'll recall that the FBI uses about 16 loci, and so you could have a maximum of 32 bands there. One band is not enough to identify one person among all the people on the face of the earth, but interestingly, because of the variability in these regions we're copying, 16 loci is enough to pick out one person among all the people on the face of the earth. Now, the only other thing to mention here is this, that on the left-hand side here, we have what's referred to as a ladder, just like a ladder you would climb. And what we see is that the ladder contains a series of segments of DNA of known size. This one has 2 repeats, this one has 24 repeats, and we know what's in there. The little bottle on the ladder told us, and therefore we can tell approximately how big our unknown bands are based on the size of the bands that are found in the ladder. For instance, this one is 14 repeats, and we know that that's the 1, 2, 3, fourth one down, so we know by looking at our information on our ladder that it has 14, that this particular band has 14 repeats. We notice that these unknowns are a little bigger. We know this is about 16 because it's a little bigger than 14. This one's about 12 because it's a little smaller than 14. So ladders are useful in determining the size of the unknown bands that we have on our gel. Again, this is gel electrophoresis. We want to be sure that we know what's the charge of DNA, what determines how quickly DNA migrates through a gel, and why would you have one band versus 2 bands found on the gel as we've just described. Here we have some other nice pictures of gel electrophoresis. We see the loading of the DNA into the gel here, and you can see the gel a little bit more nicely in this picture. Here's the negative charge, there's the positive charge. DNA is migrating through the gel from the negative to the positive because DNA is negatively charged. And there's a more realistic DNA fingerprint. The longer fragments don't move very far, the shorter fragments move a long way, and that's much what a real DNA fingerprint would look like. Here's another example. We have a nice example of DNA evidence at trial. We have our ladder here in the middle. We have a victim here with 2 bands. We have fingernail scrapings that were taken from underneath the victim's fingernails, and then we have suspect 2 shown here with 2 bands and suspect 1 shown here with 2 bands. Obviously, if these 2 suspects are the only possibilities, then the DNA is, of course, a perfect match for suspect 1. In other words, suspect 1's DNA, if there are only 2 options, must be underneath the victim's fingernails. So again, hopefully that was clear, but here's the victim's DNA, so we want to make sure that when we took that DNA from underneath their fingernails, we were not getting their DNA. And so here, this clearly indicates that underneath the victim's fingernails, we had suspect 1's DNA, which would be, a likely place to look for DNA. If someone is trying to defend themselves, then it's very likely that you would have at least small amounts of skin cells found underneath their fingernails. In the second part of our lecture, we'll talk about stem cells and cloning. What we notice is this, that every cell in your body receives a complete copy of your genome. And so through the process of cloning, every cell in your body has the potential to become a complete organism. You have the information in every cell in your body to make another you. And that's something we talked about back in chapter 11 as well. It's an interesting idea in biology, but through the process of gene regulation, cells do differ, but each one has a complete copy of the DNA. Cloning uses this potential, and we have 2 types of cloning, reproductive cloning and therapeutic cloning. They're very similar with one difference towards the end of the process. In March 1997, it was a big deal when Dolly, the first sheep, was cloned. Dolly was cloned using a process referred to as SCNT, somatic cell nuclear transfer. And somatic cell nuclear transfer works in the following manner. If we look at the pictures from our text, shown here on the slide, what we notice is this. We need some cells from Dolly, and they could be any cells that are found in the body. In the case of, Dolly was the clone sheep, right? So we're talking about the clone donor here, the sheep that was used to clone Dolly. So in this case, we took some cells from the udder of that sheep, we placed them in a petri dish, and caused them to go into G0. You'll recall that G0 is the state in which you find your brain cells and your heart cells. It's a state in which they're not going to divide anymore. So it's fairly easy to get cells into G0. At this point, we take a nuclei from this G0 cell and pull it out, And then we need an egg from a sheep. This is from any old sheep. And so we take that egg and we suck out that nucleus. The egg in this case, of course, the nucleus of the egg is haploid. Then we take this diploid nucleus. Remember, this nucleus is diploid because it came from just a regular somatic cell of the sheep we wanted to clone. We implant this nucleus into this egg, and now we have an embryo that looks much like a fertilized, much like a zygote, much like a fertilized egg, because this nucleus is diploid. We treat it with a little bit of electricity, causing it to start dividing, and now we have an embryo. And so at this point, the process is pretty easy. We take this embryo, we implant it into yet another unrelated sheep, a surrogate mother. It grows to full term, and then the clone is born. So we had 3 sheep involved in this process. We had the cell donor, we had the egg donor, and we had the surrogate mother. The clone that's born is, of course, genetically identical to the nuclei donor, the cell donor. The egg donor and the surrogate mother are just part of the process, but they have nothing to do with the DNA of the actual clone. So this is referred to as reproductive cloning. Reproductive cloning is extremely common in the world today. Now, it's not done for humans, as you realize, because of ethical concerns, mainly related to, well, I shouldn't say that. One big ethical concern is the fact that the clones may not be exactly like a normal sheep as a result of the fact that the DNA that's placed into them is a bit older. And so there's some aspects of that our book doesn't talk about. But cloning is extremely common in the world today. And so you can have your pet cloned if you so desire. Barbra Streisand has a couple of cloned dogs from a dog that she really, really liked that died. Agricultural animals are cloned extremely frequently. We clone sheep and commonly today, and cows. And I remember a student a couple years ago came back from the I think it was 15, it was a while ago, I suppose, Wisconsin Dairy Show, and he told me that the winner of that show was a clone, which I looked up and which was true, interestingly. So it's not at all uncommon to clone animals today, and it's not especially difficult, although it remains fairly expensive. Now, the second type so reproductive cloning. So there's the winner of the 2015 Dairy Show. I believe her name is Apple 2. Interestingly, we're working on cloning woolly mammoths to bring them back to life. So this is a bit of an old article here, but it is something that's being done. It's not possible for dinosaurs, unfortunately, since they've been gone for 65 million years. The DNA just isn't present. But mammoths, it is present. Mammoths have been gone for apparently as little as 7,000 years, and we have mammoths that are found in ice. The DNA is degraded, which has made things a lot slower. They're trying to patch it up with elephant DNA. But they're trying to use, they'd use elephant eggs, of course, and are in the process, humans are in the process of attempting to clone woolly mammoths. Now, therapeutic cloning is a little bit different, and so therapeutic cloning works exactly the same up to this particular point, but at this point we have things referred to as embryonic stem cells. And so embryonic stem cells can be used to create well, they can become anything. And so if you cut them in half, then you would have had 1 sheep, but you have 2 sheep as a result of splitting them in half. Or you could make a sheep nervous system, or sheep blood cells, or sheep muscle cells. And so this is therapeutic cloning. Therapeutic cloning is the process of using the process of cloning to create stem cells for therapeutic purposes. You're not creating an actual organism, but you're creating part of an organism that can be used to treat organisms that are alive today. Now, those are embryonic stem cells. Now, one thing we want to point out is this, that we have another type of stem cell referred to, this is not directly related to cloning, but talked about by your book. So the other type of stem cell is referred to as an adult stem cell. An adult stem cells, a good example would be your bone marrow. The cells in your bone marrow divide throughout the course of your life, creating additional blood cells. They can't create muscle cells, they can't create brain cells, or any other type of cell in the body, but they can create white blood cells, red blood cells, and platelets. And so those are adult stem cells. They're cells that are fairly differentiated. They have gone down this road, and they can't turn around on that road. But they can still become a couple different types of cells. Another good example would be the bottom layer of your skin. The types of cells found in your skin are created by cells in your stratum basale that divide and then make their way outward. And so adult stem cells are limited because they can't become anything, but recent studies have indicated that if we can modify the DNA in those adult stem cells, we can make them much more like embryonic stem cells. So therapeutic cloning is the process of creating stem cells for therapeutic purposes. Now, close to the end of our discussion here, we want to point out that transgenic organisms in our world today are quite common. We use transgenic organisms in medicine and agriculture. Interestingly, a large percentage of the plants, but not animals, that we produce today in agriculture are genetically modified. We do have lots of genetically modified animals, but it's a big process to be approved for food consumption. And so there's only one at this point in the United States that's approved for food consumption, and that is the salmon. And so this genetically modified salmon was approved, was it in 2016 for human consumption by the Food and Drug Administration? And so this is a salmon that has multiple copies of the growth hormone inserted, and so it grows much more rapidly than does a non-genetically modified salmon. We also, as we mentioned before, use transgenic bacteria to produce things like human growth hormone and human insulin for medical purposes. And then lastly, over here we have a tobacco plant that's been genetically modified. If you're keeping up with the news, you have read about the biotechnology company in Owensboro, Kentucky, that is one of the many companies that is making plans to produce a vaccine for COVID-19 using transgenic plants. And in this case, the Owensboro plant is planning to use a tobacco plant a genetically modified tobacco plant to produce the COVID-19 vaccine. They were in the news quite a bit. It's been several years ago now, because they produced a treatment, not a vaccine, but a treatment for Ebola using genetically modified tobacco plants. All right, as we continue our transgenic plant and animal discussion, what we see is this, that as I mentioned, a large number of our plants, but not animals, are genetically modified today. Here we see a chart that goes up to only 2014, but we see in 1996 all of our major crops. So on the graph we have soybeans, cotton, and corn. So in 1996, there were less than 20% of all these were genetically modified. We see in 2014, 80% of all these were genetically modified. So it is true that as you look out at a cornfield today or some soybeans on your commute, most likely those are genetically modified plants. Why do we do that? Well, there's two reasons. One is that we want to make them resistant to insect pest, which means that we have to apply fewer insecticides to the plants, which is good in terms of human exposure, cost to the farmers, and exposure of the environment and us perhaps in the environment to those insecticides. The second reason is that many of these plants, this is probably the most common, are herbicide resistant. Roundup resistant is the most common. And so this means that most likely if you see someone planting soybeans, they're not going to plow the ground. It's relatively unusual to plow these days. But what rather they'll do is they'll go over the field with a planter, they'll plant all the soybeans or corn in these nice little rows, and then they'll have, say, Southern States come out, or they'll have their own sprayer, and they'll spray the field with Roundup. Everything will die except for the crop that's being grown, the soybeans or the corn, most likely. And so you end up with this perfect field that has only the intended crop and no weeds as a result of the fact that the intended crop is resistant to the herbicide, whereas the weeds are, of course, not. We can get larger animals, fish, cows, pigs, from inserted growth hormone. But today, that's done, for instance, in cattle, probably swine too, although I'm honestly not certain. But in cattle, it's done as a result of implants. The cows themselves are not genetically modified. It's relatively impractical to implant fish. You're growing so many of them, and it would be expensive. So the fish genetic modification in fish, as far as growth hormone goes, is much more common. And that salmon is the only agricultural animal that's been approved for human consumption. In some cases, we use farming, spelled creatively there, to combine pharmaceuticals and the process of growing things. But the biotech company in Owensboro would be a good example of farming. They're growing tobacco, and they're using that tobacco plant to produce some sort of drug or vaccine. The COVID-19 vaccine or the Ebola drug that was produced. Another possibility is I don't think they do that at the Owensboro company, but another possibility is to make transgenic animals that produce pharmaceuticals in their milk, which would be then fairly easy to extract. Now, the topic of transgenic organisms is one that's very troubling to scientists, and scientists, I think this is probably a universally held opinion. Scientists blame themselves for this shortfall. But there's a great hesitancy, a significant hesitancy in the general public to accept genetically modified organisms, despite the fact that our best science indicates that they're absolutely safe, and there are many, many benefits to these, especially in parts of the world where people actually don't have enough to eat. We can produce, for instance, crops that can grow in areas where they would not otherwise be able to grow. And so that's a big focus of science today, is to bridge this communication gap with the general public and try to explain that, for instance, if we think back to our earlier example of insulin that is produced is genetically modified, and that's the only way that it's produced. And is there any difference between that genetically modified insulin and natural insulin? The answer is no. There's just not. So to be suspicious of it, is fine, I suppose. But there is no, to be opposed to using it really wouldn't make sense, because there's no difference between the genetically modified produced insulin and the insulin that's produced in a normal human being. It is a big topic today, and one that's interesting to look at. Hopefully that communication gap will be able to be bridged. Guys, we finish up our discussion in Chapter 12 by talking about the Human Genome Project. And so the Human Genome Project was a 13-year effort headed by Francis Collins, shown on the right here, who's currently head of the National Institute of Health in the United States, probably the most prominent scientist in the world. He was appointed there by Obama in 2009, long after the Human Genome Project was completed. And also Craig Venter, who's shown here on the left, another prominent scientist who's head of his own company, Solera Genomics. And so when they started this project, it seemed like a staggering task. In fact, in Francis Collins' book, he says he didn't see how they could possibly complete this, given the fact that his own lab had worked for years just to find the sequence of a small number of genes. But interestingly, technology got better and better, and by the time it was done, it really wrapped up. It did wrap up ahead of the projected completion time, and it was completed in 2003. So it's also interesting to note that, how's this for advances in technology? The first sequencing of that human genome cost $3 billion. But today, in 2020, you can have your entire genome sequenced for less than $1,000. And predictions are that in a very short period of time, it'll be just $100 or so to have your genome sequenced. And that's all a change in the technology. So we're not sequencing in the same way. At all. Technology has gotten so much better. The processes are completely different now than they used to be. So the Human Genome Project changed everything as far as our knowledge of science and medicine goes. I was watching a movie last night that had Angelina Jolie in it. Angelina Jolie, of course, is probably the most famous person I can think of that has been positive for this gene referred to as BRCA1. BRCA1 came out of the Human Genome Project, and Collins talks about it. I think it may be in his second book called The Language of Life. He talks about this very specifically. So in other words, they're sequencing away in the Human Genome Project, and they come across this particular gene, BRCA1, and they realize that if you have this gene, it's almost certain that you'll develop breast cancer during the course of your life. And so they made that sequence available almost immediately to some families that were in a trial. And he talks about one example of a woman who was going to have both her breasts removed, but as a result of the information that came from this sequence, she realized she didn't have to have that surgery at all. She was no more likely to have breast cancer than anyone else, simply because she had not inherited that particular gene. Unfortunately, Angelina Jolie had, and she wrote an article in the New York Times a couple years ago about the fact that she did have a double mastectomy to prevent her from developing breast cancer. And that's just one little example. It takes 20,500 genes to make up a human being, and that's one of those 20,500 genes. Now, that leads us to this discussion of proteomics. And so proteomics is the study of proteins, of course, products produced by these genes and what they do. This is so important in drug development, and this is a lot more complicated than the Human Genome Project. So sequencing DNA is pretty quick and pretty easy and fairly cheap today. But figuring out what those genes mean, in other words, what proteins do they produce and what is the significance of those, is a much more drawn out process. And more information is added each day in terms of determining what is the function of the genes. So to be absolutely clear, hopefully, proteomics is the study of proteins, and the term proteome refers to the entire collection of proteins that's produced by an organism's genome. As we talked about in chapter 11, the proteome and the genome, it's not a one-to-one correlation. Because of alternative splicing, you can have a quite large proteome, which is the case for humans, even though the genome is not really that large, meaning that the number of genes is not a good predictor of the complexity of the overall organism.